There are about 3 or 4 of these vocabulary words which you will not have to know. An ester bond, you will not have to know it for the exam. It's probably a good idea to know what an ester bond is as you're reading it, but it's not necessary. So let's go down and check off the ones you should know. Amino acid, for sure you should know what that means. Allopathic, you'll know after we discuss water. Carbohydrate, you should know. Carbon, you should know. Denaturation, you'll have to know when we're done. I'm going to put a little asterisk if we haven't covered it yet. Disaccharide, you should know. Fibrous protein, you should know. Globular protein, you're going to need to know. Glycosidic bond, man, I'm not sure, but we'll go ahead and mark it. We've already talked about it, you just it hasn't been named. Hydrogen bond, you'll need to know. Inorganic ion, you'll need to know. You already know those. You know what an inorganic ion is. It's a charged atom or a group of atoms that are not organic. In other words, not carbon-based, right? Right? Because that's what organic means, is carbon-based. Lipid, we're going to talk about today. Macromolecule. And it's just a large molecule. Again, we'll make sure to define it. Monomer, you should know already. Monosaccharide, you should know already. Nitrogen, you should know already. Nucleic acid, you should know already. Nucleotide, you should know already. Organic molecule, you should know already. Peptide bond, you should know already. Phospholipid, we will learn today. Phosphorus, you should know what that is. Phosphate as well, right? Polar molecule, we're going to cover today or in the video that I'm going to send you over the weekend. Polymer, you should know. Polysaccharide, you should know. Protein, you should know. Surface area volume ratio, we're not going to cover today. Not going to be on a test yet for this unit. And water, you should know. Now, you think, oh, water. There's actually there's a lot about water you don't know that you will learn. So we're going to go put an asterisk by it. So half the stuff in this packet you have already done. You say that's funny that water's wet. Well, actually, water's not wet. So anyways, whether you're smart aleck or not, you need this, half this stuff you have already covered. So you should be able to get through at least half of this quickly. As you look at the key concepts, you'll see that organic molecules are carbon-containing molecules. They're central to living systems. So if you need to know these highlights, these are the main ideas. Water and its properties we will probably deal with on Tuesday. As you're going through this, you should be able to check off each of these boxes. It's a checklist for you to go through as you're trying to figure out what is it you don't understand and what you do understand. Do I understand all the key terms in the glossary? Well, how do I know? You have to check. You have to check yourself. You have to actually check to see if you know it or not. And how do you do that? You quiz yourself. You have to identify the common elements found in organisms and give examples of where these elements can occur. You know this. Schnapps are the common elements found in living systems. And you need to know more than this packet's telling you, right? More than this study guide's telling you. You need to know what elements are in lipids, what elements are in carbohydrates, what elements are in proteins, and what elements are in nucleic acids. You need to know it, you should know it, and this is the video I already sent you. So really, you should know this already, and I know it, I can check it off. And so you could go down the list and check off whatever it is you don't know. And if you don't know it, maybe that's something you better make sure you come to a tutoring session on, or maybe that's something you want to make sure you take care of before when? Next Friday. If you can do it before tomorrow, that'd be great, because then you can come in on Saturday tomorrow and get some help. This is fairly easy. I shouldn't have to explain to you. You need to read this. Look at these key terms. This idea of biological molecules, nucleotides, nucleic acids, of carbohydrates. Where are they found? How are they used in living systems? How is water used? What are lipids? How are they used? You should be able to do that as you're looking at this and you're going through this. We haven't covered the cell yet, but this is your introduction of where you find these biomolecules in cells. And although water is not really a biomolecule, it is a molecule that's in life. Proteins, and where is it that you find proteins? It's not an organic molecule, rather. All right, I shouldn't have to explain this. I think you could read and you can explain this yourselves. And you should certainly be able to do on your own, right? Yes. You should be able to quickly tell me what all these do. I mean, I don't know what else to say. If you can't do that, you have a lot of work to do. We've covered it in class for the last few days. And there's videos if you need to. Role of water we'll come back to. So if we don't get to it today, you can come back to it next week. Or you can do it on your own. You can probably read and figure it out on your own, honestly. But it's up to you. Organic molecules. What are organic molecules? What are functional groups? I think you should be able to know. You know some functional groups already. What are some functional groups you know? Amino. You know that group, right? Yes. You know what an amino group is. You know what a carboxyl group is. And you know what a hydroxyl group is. So these examples we've already covered in class. You should know what a macromolecule is. A macromolecule is something like a protein or a cellulose molecule, large molecule, bunch of monomers usually, but it could be even lipid, giant molecule all bound together and making something. What it is depends on what is being held together. Here's a molecular formula. We've already covered molecular formulas. Here's an example of a molecular formula here. Structural formula, that's what this stuff is here. This is the structural formula. We've done them before, haven't we not? We've also done this structural formula here. Actually, this is the one. These are both structural formulas. We've done both of them. There's no reason that you shouldn't recognize what they are. We've drawn them in class. We've literally spent hours doing this. Models. What are different models? This is different models. You see me write out C6H12O6, and you see me draw it out this way. Well, there's different ways to represent the same thing. All of these are the same thing. They're just drawn out in different ways. Each one describing a different aspect of that thing. In this case, it's glucose. Here are some functional groups. Here's some. We know carboxyl, right? This is the one we talked about for carboxylic acids. There's also something called a ketone and aldehyde. Do you have to know this for the tests? No. No, you do not. Should you read it and be able to answer these? You should be able to answer these questions based on this reading. So yes, I expect you to answer it, but do you need to know it for the test? No. All right. You should understand that these things called functional groups. Functional groups, you should know what they are, but I'm not testing it. I'm not going to put on a test, what is a functional group? Give me an example in a functional group. But I'm going to tell you what. If you can identify a carboxyl group or an amine group, it's going to help you a lot. Because I will ask you, which is this? A protein, a sugar, a carbohydrate, a lipid? And if you know what a carboxyl and an amine group are, you can identify the amino acid. So, yes, you should know some things. You should at least know these three, right? All right, moving on. Building an, here you have where are all the different elements? Carbon, and you can find them where? In all the biomolecules. Carbon, you are a carbon-based life form. All life on Earth is pretty much carbon-based. Carbohydrates, proteins, lipids, nucleic acids, all four of them are carbon-based. You should know that by now. There should be no mystery there. Phosphorus is, you should know that phosphorus is found in lipids, but it's mostly in nucleic acids. Oxygen, how do you use oxygen? To breathe. It's in the atmosphere, use it to breathe. Carbon, the source of carbohydrate, again, back to looking at that. You should be able to identify all these, right? This is not difficult. I'm not going to go through it. These are ions. They're charged elements, charged atoms, or groups of atoms. Is this organic? Do you see carbon? If there's no carbon, it's not organic. Write that down. If it is no carbon equals not organic. Another word for not organic is what? Inorganic. Inorganic. So these are all what? Inorganic. There's no carbon in any of these. These are all ions. All of these are ions. They're all important for life. These, you need to know these four, all these inorganic ions and what they do. You need to know them. It's not hard. All of you know that you need calcium. Do you not? What do you need calcium for? Bones. Bones. Thank you. Teeth, bones, muscle contraction. Actually nerves as well to some extent. So bottom line is hearing actually is very important, calcium. But in any case, you know this. This is something you've been learning since you watched your milk commercials on TV. Magnesium. You should know it's in chlorophyll. How do you know? It's here. It's on the chart. Figure it out, answer it, put it on index cards, memorize it. You got a week to do it. Not hard. Honestly, I don't know what to say. Okay, biochemical tests. We will be doing this test, this RF values. This is our lab for next week, so make sure you read it. Make sure you read this and how we do this, because we will be doing chromatography next week. You'll see it, you'll do it, I'll give you instructions, you'll write it, you'll take pictures of it, you'll make your measurements, you'll calculate your RF value, and we'll go on. Know what these three tests are. This is, again, memorization stuff. This is stuff that you either know it or you don't know it. It's not really hard. How do I know if there's iodine in something? I'm going to use an iodine test. How do I know if there's sugar in something? How do I know if there's proteins in something? Each of these test a different biomolecule. So these are different tests that you should know. If you want to find out if you have protein in something, you're going to do the Barrett test. If you want to find out if there's starch, you're going to do the iodine test. If you want to find out if there's lipids in something, then you're going to do the emulsion test. Do you want to know if there's sugars? You do the Benedict's test. Each test tells you something different. You need to know this because on the following week, we're going to use these tests to test different substances. So you need to know what each of these are. There should be no surprises in 2 weeks on which test tests what material. Is this going to be on the test on Friday, on your exam on Friday? Are these tests going to be on the exam on Friday? No, not on exam. Do you need to know it for the following week for your labs? Yes. Needed for labs. In 2 weeks, you need to know what they are and how they work. This will be on next week's lab. And honestly, once you read it and look at it, it really isn't that hard. So this is going to go over the procedure. It talks about the background. You need to do this before Tuesday because your lab will be Tuesday, right after your quiz. What am I going to quiz? I'm going to quiz anything in this anything in this thing is quizzable. Is testable. My leaning is to quiz this stuff because this is what the lab is going to be on. So, all right, and so again, you have some questions here that you can look at, explain why the R value of a substance is always less than 1. You can go ahead and write those out and think about them. I'm not going to tell you the answers, but I have confidence that if you think about it, you think about how this works, you should be able to come up with an answer. This is 100% review. Nucleotides, nucleic acids, everything in here we covered. There's a whole video on it. It's already been posted. It was posted 2 days ago. I love this. This is much better, much neater than the drawings I put together in the video for you. It talks about a base, nitrogenous base, sugar, and phosphate forming the nucleotide. It does links. It shows you clearly the RNA molecule, the DNA molecule. It shows you a different picture of the DNA molecule than the one I drew. Really well done. There's nothing here, absolutely nothing here that you already don't know. If you don't know it, you need to read it. And if I were you, even if you knew it, even if you're like, I know that, I'd read it anyways. Because what are you doing when you're reading it and taking notes on it? Studying, reviewing and studying. And write down, take notes, and revise these things. So this is how you study. So this is how a nucleotide is formed. And I want to point something out. In a lot of your chemical reactions, not most, but a lot of the chemical reactions that are going on in your body to make DNA, to make carbohydrates, to make carbohydrate polymer, whatever, you're constantly making water. Every time you put something together, you're making water. It's called a condensation reaction. We're going to look at it in a minute. But you see water's always coming out. Look at it. Hydrogen and oxygen get kicked out, and the two things are linked. It's called a condensation reaction when water is removed. So you should know that. That will be on the test. That is testable. Double-stranded RNA, or DNA, what is it? There it is. That's explained. Again, I love their drawing. I think they do a much better job than I did when I drew it. This is DNA. There's the backbone, sugar phosphate backbone. Here's a sugar phosphate backbone here. Here's the nitrogenous bases. These are nitrogenous bases. There's the hydrogen bonding. These are the base pairings, ATGC. Do you know which this is? Is this a purine or pyrimidine? Is this a purine or pyrimidine? Is this a purine or pyrimidine? Pyrimidine. Is this a purine or pyrimidine? Purine. Double ring purine. Single ring pyrimidine. You should know that by now. It's 2 days ago. Well, I forget. I didn't do it. Look on the previous page. And you'll see there they are listed for you. Again. Study guide. What are the differences between DNA and RNA? I can guarantee you that's going to be on the test. If you can do this without looking and reading, if you can answer these, you're ready for that part of the test. If not, you better get to moving on those index cards, whatever it is you have to do to study for that. Amino acids, we talked about it, didn't we?
Yes.
Yes, we did. And we looked at the structure of amino acids. There it is. NH2, the amine group. C double bond O, OH, carboxyl group. There's the hydrogen atom. There's the center carbon. 2 carbons. NCC, 2 carbons. There's the R group sticking out. Hydrogen. All right, the center carbon has a side chain. Well, wait, what does a side chain look like? Well,
There's like the carbon model that we made the other day, yesterday.
That's the art block.
that is, this is the stuff we did yesterday.
Yep.
And there's 120 amino acids in your cells, but only 20 are found in proteins. And so those 20 are the ones we're going to focus on. 20. Those 20 have different side chains, and each one has a different quality, chemical property. Some are alkaline, which means they're basic. Some are acidic, right? There's a C double bond, COOH, carboxyl group. Here's a sulfur. So what can that sulfur do? The disulfide bond. Remember we talked about hair in the last video and how the disulfide bond can form. So that bridge, the disulfide bridge. And there's disulfide bridge. There it is. Again, review, right?
Yes.
A review. And here's these drawings, much better than the drawings I drew. Much clearer. Here's cysteine, here's lysine, here's aspartic acid. Notice that these three are all the same except for what? Except for the R group. When they're linked together through condensation reactions, we form water, we link them together, they're the side chains, forming this polypeptide chain. What do these do? Oh my goodness. Room 374, Mr. Mendoza speaking. May I help you? Let me see. Hold on a second. I'm in. All right. So when we look at these different amino acids and where they occur, you see that these. You should read these and find out where all this stuff happens and where they all occur. It's all very important. But it's all a review and an application of what amino acids are. Nothing new there. Nothing new. This process, this condensation reaction, I literally showed you 2 days ago on screen and recorded it and posted it. This condensation reaction, this linking between and water coming out, I showed it to you. I drew it. I didn't call it condensation, but there it is. There's a name for it. Condensation reaction. You have to know it. You have to know what it is and how it happens. There's example after example of it. What you may not know is that once you have it connected, you can unconnect it by adding water to it. You can disconnect it by adding water to it. In any case, you can go ahead and read these and write down anything you have problems with. You might want to ask me. When? During advisory. When? During tomorrow. When? After school on Monday, Wednesday, or Friday. Not Friday, not today, because I'm not staying after. And then next Friday, it's a test. It's too late. You can stay after and ask me anyways, because there'll be another test covering the same material. But you know this stuff. Fibrous proteins and globular proteins. There's the definition. Remember we said in the beginning, vocabulary words that you need to know. There it is. Once you have them put them on index cards. On one side put fibrous, on the other write these qualities down. Globular proteins. On one side put globular protein, on the other side put this down. Know what these are. They will be on the test. And they're exactly what you see. Fibrous proteins are found in structural stuff, and stuff that's gonna be used to support you. Your skin, for instance, is collagen. There it is. They're fibrous. They're fibers. Your skin is made of a bunch of fibers and proteins all interlinked, just like the hair that we talked about yesterday.
Are your organs made of fibers?
The stuff that's called connective tissue, the stuff that holds your cells together, the cartilage in your nose and your ears, the tendons, the blood vessel walls, they're all made of these fibrous proteins, these structural proteins. The globular proteins they're water soluble, they form things like enzymes. We're going to talk about enzymes. We talked a little bit about them yesterday. When I post the video, you're going to see that there's another 10 minutes after we finished yesterday that talks a little bit about enzymes, so you really need to pay attention to that. Catalytic enzymes, enzymes make reactions go faster. You want to know more? Watch that last 10 minutes of the video that I'm going to post today. Not this one, the other one I'm going to post today. This one will be posted today as well, by the way. Regulatory hormones, you know what hormones are? Things like insulin, estrogen, testosterone. These are all hormones. They travel through your body. They are globular proteins. They have to be soluble in water. And protective antibodies are also. These are all examples. Of course, there's many more. Look at this. Look at what we find in between these proteins. Cysteine and cysteine. What do we find? The disulfide bridging or disulfide bond that we talked about before. Over and over again, you see the same stuff. Do you really need to know beta-pleated sheet or beta chain? I'm not really concerned about that. That's something that you might need to know in the future. Certainly for AP biology, you'll need to know it. But the key is that you understand that this chain of amino acids, on one side, every time you have two cysteines, they form a bridge, which does what to the protein? Folds it. It folds it, bends it. This is insulin, by the way. This is insulin in a bovine means cow. The word bovine means cow, in case you're confused. Nice. I should say your name so that your mother knows who you are. Mooing in class is not cool. So anyways, try to answer these questions. You should have no problem answering these. Let's go through and make sure we understand what these are. Structural tissues, you should be able to identify that. How are the proteins involved in that? You read, it talks about it. Regulating body processes, right? That would be things like hormones. You should be able to contractile elements. Those are going to be things where you have muscles contract. Things like antibodies are involved. Transporting molecules within cells and bloodstream. That would be hemoglobin. Catalyzing metabolic reactions. That would be enzymes. Denaturation and effect on protein function, we're going to have to talk about. Still haven't spoken about that. So let's talk about that now. Fibrous proteins, you should know why they're important. If you read, you should be able to tell why they're important. All right, so let's talk about it. First of all, let's talk about regulation. Proteins act as signals. They act as signaling molecules. Signaling molecules, they go from cell to cell telling a cell what to do. So that's how you involve yourself. Those are an example of signaling molecules are hormones. Hormones help regulate your body. Structural components, structural tissues help support you. Skin keeps you separate, the parts and pieces separated. Connective tissue holds your organs in place. How would you like it every time your heartbeat jumped around in your chest? That wouldn't be any good. You got to hold that thing still. That would freak me out if every time my heartbeat moved. My chest just started thumping. So obviously you should be, those two I think are covered now. Contractile elements. Listen, when you have these fibrous proteins and they connect, then you could do this. You can slide this fiber can slide this way and this fiber can slide that way, then whatever's attached over here and whatever's attached over here can do what? Bend. Move closer. When you bend your arm, that's what you're doing. When you move, all you're doing is taking fibers, muscle fibers from one side, muscle fibers from the other, using this connector in between, and you're sliding the fibers, you're ratcheting them closer to one another. So the two ends of the cell are moving closer together. These cells are attached to bone, so the bones move. That's how, and you do that using proteins. Immunological. Immunological means what? Immune system, that's right. Immune systems. And you have these things called antibodies. Antibodies are what? They're proteins. They're made when something foreign enters your body. And what do they do to the foreign thing? The bacteria or the virus? They attach to it, and yes, that's right. The white cells, the white blood cells attack it because the antibodies are attached to it.
You said antibodies are proteins?
They're proteins, yes they are. Proteins made by immune cells in your body. Transporting molecules within cells and in the bloodstream. Well, isn't that important? Don't you have to transport oxygen from your lungs to your tissues?
Yes, it was.
So hemoglobin's an example of that. Hemoglobin, globin molecules, globin, right? Hemoglobin is very good at carrying oxygen. It's very good at carrying oxygen. So if you're going to carry oxygen to your bloodstream, it better be what? It better be soluble. Let me rephrase that. Yeah, that's right. It better be able to dissolve in water, right? Because what's your blood make? What's the solvent in water? Water. I mean, the solvent in your blood is water. That's what I meant to say. Is what's the solvent in blood? So whatever it is that's going to carry something in your blood, it better be able to dissolve in water, because your blood is made, the solvent that's in water, that's in your blood is water. So if it's going to be suspended in your blood, it has to be able to be suspended in water. That's why sugar and salt can be in your bloodstream. How can it be in your bloodstream? It can dissolve in water. Explain denaturation. Okay, this is kind of a simple idea, but it's a very important idea, so pay attention. This will be on the test. This is absolutely testable. I'm going to put a little red asterisk by it. You know what? Let me get rid of this and just put a red one. If you have a protein that has a shape, let me draw a better shape. If you have a protein that has a shape, it has to have this shape in order for it to work. It's going to work on a substrate. Remember, enzymes and substrate. It's going to work on this substrate. In order for this to fit into this, this has to be this shape, this has to be this shape. Is that understood? Mm-hmm. That makes sense, right? Yeah. Like a lock and key. If you increase the temperature, remember what happened to the hair when we did a flat iron to the hair? Remember the disulfide bridges broke, but the protein stuff, the primary structure stayed the same. So it changed shape, right? Yeah. Well, that's true of every protein in your body, including your hemoglobin and your collagen, your skin. When you heat it up, this molecule can change shape. If it changes shape enough, then it no longer fits. Then the substrate no longer fits. If the substrate no longer fits, then that reaction stops. It doesn't work anymore. What we call that is death. You die. The result of that, if it happens for a long enough period of time, is death. Well, what is it? What are the things that change the shape of this protein? obviously heat, right? Because we just talked about it. So obviously heat is one of those things.
So that's why we have like heat stroke.
That's right. Good. I was getting there. Yeah, that's where I was going. That's why if you go, what is it that, what we call denature? Why do we call it denature? Because there's a natural form to the protein, and excessive heat, right? Or salt can do it too, or pH, high or low, can all change the shape. All of these can change the shape of the protein. And if the shape of the protein changes, it doesn't work anymore. Which means the things that make you do what you're able to do, like breathe, like think, like carry oxygen, for instance, if you unfold hemoglobin, then it can't carry oxygen. If it can't carry oxygen, you're dead. So if you go into a hospital with a high fever, it happens. Mr. Y.
I didn't know. I didn't say nothing like
I thought you were making fun of me because I misspelled something.
I was.
I saw that. Oh, I saw it. Trust me. I marked it. It's a tick in my brain. All right, so fever. High fever. You go in the hospital, take a baby in the hospital, take a person in the hospital, high fever. What do you think they do to you right away?
No, they get for the chocolate.
They put you in ice water.
Ice water.
An ice bath. Actually, they have better ways of doing it now, but if in an emergency, they'll put you in ice water. And the reason they're doing it is they have to bring your temperature down, because what's happening? Your proteins are what? They're not melting, they're unraveling. They're losing their shape, just like your hair is losing shape. If we can cool you down, then what will the proteins do? Go right back to their original shape, just like the hair that we discussed yesterday, right? Assuming you didn't overheat it, that if you get burned, you've changed the shape permanently because you've broken the covalent bonds. But what we're talking about here is those disulfide bridges and hydrogen bonding that occurs in these proteins. If we can keep the fever down, keep the temperature down, we can let the proteins go back to their original shape. Yeah?
So some people don't all go to the hospital like when they get a fever. So what do you do? Like what's the medicine that they take?
It's a good question.
What's in it?
A fever, a low-grade fever, it will not do this. It does do this to cells that are more sensitive. And so the reason your body gets a fever is because the bacteria or the cells that are invaded by viruses in your body, they're sensitive to heat. They're much more sensitive than your cells. Your cells are much more complicated and much more protected. So if when your body gets a fever, it's killing the bacteria. So the fever is not actually it's a good response to a bad thing that's happening to you. You're being invaded by some creature. And that fever is helping protect you.
Oh, that's why you use it.
That's why you get a fever.
Okay, but what's in the medicine?
Now, when you get medicine, there's medicines that'll signal your body, stop. And I don't want to talk about it exactly. Acetaminophen, for instance, Tylenol, does that. It helps reduce your fever. But sometimes the fever, your response is so extreme that medicine doesn't help. And in the short term, the only way we can get your fever down is with ice. Yeah.
If the protein shape changed, will it be able to be fixed?
Yeah. As soon as you reduce the temperature, assuming you didn't break the covalent bonds, right? It'll do something. Here's the word. You ready? It'll renature. So denature, it unravels the change shape of protein. Renature goes back to normal. Is that understood? Yeah, question?
You know how you were just talking about fevers and stuff?
Uh-huh.
How does, like, when you take a pain pill, how does the medicine know where the pain is in your body?
You know, when I was a kid, I used to ask that same question. It's a really good. I think it's a good question. What it doesn't. The medicine affects all your nerves.
It's just the
All your pain receptors are all impacted by that medicine. So if you have a toothache and you're taking aspirin or whatever, your toothache might feel better. Well, if you had back pain, that would feel better too. It affects all your pain all throughout your body. So why are fibrous proteins important for structural molecules both within cells and extracellular? Guys, fibrous proteins hold you together. Skin look, it's right there. It holds you together. Fibrous proteins hold together your cell, give your cell shapes. And it suggests why globular proteins will contrast fibrous proteins have a catalytic or regulatory role. Because they dissolve in water. If they dissolve in water, notice what they can do. These globular proteins can dissolve in water. If they're going to work in your blood or work in a cell, they have to be able to work in water. Fibrous proteins are what? They don't work in water. They're good for protecting and holding things together. Yeah.
Okay, so when you need like fiber bars, those help you like.
Fiber bars are different. It's a good question, but it's a different situation. I'd rather not talk about it now. They're not soluble in water, so what they do is you can't digest them very well. And those fiber bars, what they do is they help clean out your intestines. Because water comes out of your body and into your intestines, and it washes out your makes things go faster. This is a little more complicated, so pay attention. So these kinds of proteins, after we translate proteins, after we've made proteins, after these translational means transcripted and translated, that means we've made the protein, right? And ribosomes are enzymes that make them. We can make what are called glycoproteins. We can take the sugar. What does this look like? All right, we can take these groups of sugars, and we can attach them to what? The protein. And together they make a what? Glycoprotein. So together they can form glycoproteins. So we can cleave these polypeptide chains after we've made them. After we've made them, that's what post-translational means. After they've been made, okay? Right? Read it, and it says proteins may be modified after they have been produced, right? After they've been made. We call them post-translational. We can make glycosylation. That means you're adding sugar to it. You can do phosphorylation. What does phosphorylation mean? What are you adding to it? Phosphorus. Phosphate, right? Phosphate groups. That's phosphorus with oxygen. You can attach a lipid to it. Look at it. You can attach a lipid to it, which will allow you to put it into the membrane. That's a lipid, by the way. To what? To the protein. I don't know why you're going in your bag. I'd like you to pay attention. And you can do degradation. You can break down. You can take and break the protein down. So these are all functions you can do with a protein. And so it's important you get these functions straight, because in the next unit, the very next unit, we're going to start making DNA and making proteins. We're going to actually put them together, and we're going to look at a cell. And we're going to put together a cell, and we're going to ask ourselves, how do proteins and lipids and carbohydrates all fit into a cell, and how do they function? And we're going to go very quickly. It's going to go very smoothly, hopefully, but only if you know what all these terms are. So read, understand, and answer. So here again, the biological forms of lipids, you can take a look at these. All right? Obviously, they're for energy. You can read it. You know that fats and water don't mix, right? All right, we'll talk more about water later. There's these fats, there's oils, there's saturated, unsaturated. Remember I said fats or lipids? Lipids is a big term for fats. They're long chain of carbons and hydrogens with an acid at the tip. This is glycerol, a 3-carbon sugar that can take on They can take on fatty acids. This is a fatty acid. This is carbon and hydrogen. These are all carbons bonded to each other. They're long chains of carbon. This is a fatty acid right here. You can combine them into what's called a triglyceride. So you can answer these questions on your own, I think. I don't think you need me. Here's phospholipids. This is what makes up your membranes of your cells. So make sure you know how to handle that. Please don't put anything away until I'm done here. You will not have to know what isomerism is. Isomerism, all right? It's a big word. It's an important idea, but we're not going to have it on this test. Read it, be able to answer it. Not going to be an issue for this test, but you do need to know, stop putting things away. Condensation reactions, hydrolysis reaction, all right? You need to know that. You should be able to tell me most of this other stuff. It's all on the test. It's all in the videos, or rather, and it's all on the test except for isomerization. You should be able to tell. You put things away. I say sit down. I say don't do it, and still you do it. Sucrose, maltose, lactose, cellulose. These things we covered. Here's an experiment. Make sure you'll be able to do it. Here's this polysaccharides. I'm not going to talk more about it. We've already talked about it. Here's cellulose and starch. Make sure you're able to know the difference. And lastly, but not least, some more questions about structure. You should be able to handle this. All right, good luck. Put up your stools, please, and pack your stuff up. Make sure your area is clean. I appreciate it.